Oh, Greg, is that me or is that you leading off here?
I'll just say something really quickly and say, hi, I'm
Oh,
Dr.
okay.
Greg German. I'm the chair of Global Clinical Phage Rounds. Welcome to 2026. It's been a little bit of a slow start, but we got a fast start for the rest of April and likely more cases to come. We've had a great interest from our colleagues at Phage Australia. I can personally thank them a million times for all the support they've provided us, both in the Canadian context as well as globally. And Professor and Dr. John Iredell has been the key driver for that, and he's going to be um, what's the right person? The person in the middle of the three-ring circus, perhaps, or I don't know how you want to classify it, John, but we'll let you continue with the rest of the talk. And we also have Dr. Gina from the Mayo Clinic as vice chair, and both myself and Gina will be trying to assist with the monitoring today. Thank you. John, take it away.
Yeah, thanks, Greg. Thanks, Gina. So, as many would know, we in Australia do not have the same rich history of our colleagues in Europe and indeed in the US with phage therapy, but we have started introducing this recently, and we're now facing the big question of how to establish it as a potential national therapeutic strategy. So, with that in mind, and apologies from Ameneh Khatami, who was centrally involved, as many would know, with the development of the stamp protocol that we use, I want to introduce Dr. Manisha Yapa, who's an infectious disease specialist, and she's going to talk about her area of expertise and research interest in the science of implementation, which is critically important and I think not discussed enough. We have Dr. Kerry Watts, who's a senior project officer in advanced therapeutics at the Ministry of Health here in New South Wales, who's going to give us a bit of perspective about regulatory aspects that are so important in this. Carina Lauter, who I think has spoken before at this meeting, but in any case is our quality lead and biobanking lead here at Westmead. She's going to touch on the biobanking and quality management aspects. Stephanie Lynch, who I know has spoken before at this meeting, is going to speak about the feasibility of the production and the diagnostics, which of course is what we rely on to actually deliver the goods. And then, as promised, Holly Sinclair, who is also well known, I think, to this audience, will speak a little bit on safety aspects that we have had to face. So I might hand over to Manisha and I'll stop sharing. Manisha, are you okay to share screen?
Yes, thanks, John. I'll bring up my slide set.
Thank you.
Right. Okay, can everyone see my slides?
Yep, you could go to full screen.
Yeah,
It's perfect.
great. Thanks, John, and thanks everyone for inviting me to be a part of this meeting today. So my interest is in health system strengthening and health service quality improvement. So I'm just gonna give a really brief overview of how that might be relevant to scaling up phage therapy, which is, as you well know, a novel therapeutic intervention. And so implementation research really helps us to understand how best to implement generally an evidence-based intervention at a broader scale into routine clinical practice, so within routine clinical services. And this is important because not only is it important to have a good supply of phages, and by supply I mean broadly in health economics terms as well, but also from a service delivery perspective. So we might have phages available, we might have the correct phages available, and we might have physicians who are engaged or who are able to deliver phage therapy, but we also need to create demand for it. So we need physicians who think about administering phage therapy where it is clinically indicated, as well as patients understanding and being willing to receive that therapy. So with that, I will move on to some frameworks which are important for understanding I guess it gives us a structured approach to implementing a novel health service intervention. And just going back to that supply demand point, if we think about vaccines, we know that vaccines are efficacious, but they're not effective at a population level unless there is uptake. So with designing health service delivery interventions, this is now going more general. There's a bunch of frameworks out there. And what they help us do really is have a structured approach to understanding what is currently going on in health services and what is available, and then helping us use that structure to then develop service interventions. And also, once we are implementing something, to understand how best to evaluate it. Oops, sorry. So for example, there is this framework developed by the UK Medical Research Council, which is called the Process Evaluation Framework. So complex interventions are interventions which have many different moving parts. So you have people involved, it involves behavioral change. There are various resource issues, how they are supplied, what happens, and so on. And so as with any health service intervention and also in the different environments, context is always important. So if you have an intervention, then how is it implemented? What do you need to do? Who does it? How often is it delivered? Is it delivered as intended? So that's what fidelity is. Does it reach the target population? And by understanding these issues, you then understand how it achieves the desired outcomes. So in this case, health outcomes. So that's what a process evaluation is broadly, and that can be used to design the intervention itself as a framework, and then also implement and evaluate it using various data monitoring indicators. There is another framework called the Tailored Implementation of Chronic Diseases Framework, which has 7 domains and looks at things like clinical care guidelines. For example, does the intervention align with existing clinical care guidelines? Health worker factors such as knowledge and motivation, interactions among health workers such as handover or referral systems, patient factors, resources, including financial resources always, but also things like technology and infrastructure and so on, capacity and willingness for organizational change. So obviously, if you're implementing a novel therapy, that requires some level of organizational change. And then social, political, and legal factors, which may be supportive or be a barrier towards implementing something novel. Another framework which I often use in my work is this framework called acceptability of healthcare interventions, which can be used for health workers as well as consumers. Whoops. So having gathered, so the first step with implementation research really is understanding the local needs or conducting a needs assessment or a situational analysis. In other words, what is currently happening in terms of service delivery, and within that system, how are we going to introduce this novel therapy? And one of my favorite frameworks for analyzing and synthesizing all that information is the systems thinking framework for health systems. Some of you may have heard of the WHO building blocks. So there are 6 building blocks, including human resources, supply chain, financing, and governance, and so on. But that framework does not really consider how these different components interact with each other. And it also does not include the needs of the people whose health we are actually trying to improve, nor does it consider how those components interact with the broader context. So this is why I like the systems thinking framework, because it considers all of those issues. And what this diagram is hoping to depict is within that complex system, you are introducing a new intervention with desired health outcomes of some sort, and the diagram shows how both the intervention and the outcomes are influenced by what is going on within the broader health system. Importantly, though, is also how this intervention and or the health outcomes have knock-on effects on other aspects of the health system. For example, if you are drawing on human resource time to implement a new complex intervention, then those same human resources will be less available to deliver other services that are also within their job description. So that might then have a negative impact on other services, or because this new intervention is also training those human resources in certain quality aspects of care, those people might then be transferring those skills to other services, in other words, having a positive knock-on effect on other services. So this intervention may end up having a net beneficial effect on the system. Now, so that's the systems thinking and analyzing it all. But how do we actually implement an evidence-based practice into routine care? So I've already mentioned the situational analysis and understanding what is currently going on, but needs, patient needs and health worker needs are also important to understand. So designing a suitable service delivery model can be approached using this concept of design thinking, which some of you may have heard about. It is something that is quite is increasingly used in industry and starting to become used in healthcare as well. So you empathize and then you define your root cause so-called problems. For example, the gap might be health worker knowledge or training, or the gap might be in financing. But you define what that is, and then together with stakeholders. So in an ideal situation, you would get the health workers, you would get consumers, anyone and everyone who is involved in successfully delivering this intervention, get them to come together and ideate. So this is where you use your creative thinking techniques, where you diverge, you just even the most wild and unrealistic idea gets put out there. And then you converge on things that are pragmatic based on available resources and other considerations to develop a prototype model of service delivery, which you can then test, because of course you would also have to design some indicators that you're going to measure success of implementation with, not just health outcomes, but things which are within the sphere of influence of that particular clinical practice. And then you implement it and you evaluate it on a regular basis to see whether things are working as intended or whether it's been a complete disaster and you need to abandon that particular service delivery model and go back to the drawing board, or you might be able to adapt it, just minor tweaks. Now, this approach really draws parallels from continuous quality improvement, which is here I'm depicting tools from the Langley model for improvement, where you are planning what you're going to do, you do it. Of course, in the planning stage, you define who is doing what, when, and how. So there is some responsibility allocated. Then you do it, you study it, and then you act, meaning you abandon, adapt, or adopt the intervention. Sorry, my slides are flicking all over. So now bringing this all back to phage therapy, obviously it's important to engage different stakeholders involved with the process at all levels of the system, including consumers, including government, the health workers who are implementing it, and so on. And we will later on during this session understand some of the other aspects of implementing phage therapy. Then design thinking, which is how you co-design a best practice model of care and then evaluate it using process evaluation methodology. And of course, to do that, even if you're not doing a process evaluation, it's really important to have established monitoring and evaluation systems. For example, at this stage, at this early stage of implementation, probably just treatment response, but in the longer term, with more widespread use, there might be population level effects, ideally reduced antimicrobial resistance at a population level, for example, to monitor and see if things are working as intended, which you can then use to go back to the drawing board if things are going in the wrong direction, and redesign your strategy. So I'm going to stop now.
Thank you, Manisha. That was fantastic overview. What we might do is hold our questions until the end, but we might move next to Kerry, if you wouldn't mind, Kerry, sharing your slides. So we're going to think about I guess, some of the more regulatory and government-facing aspects next.
Okay, everyone can see my slides?
Yep, that's perfect.
Great. Okay, thanks, John. So thanks for the segue as well, Manisha. So obviously we're talking about health system implementation. So I'm going to talk a bit about the regulation of bacteriophage production and how we've got there over the last few years. It's not been quick. So just a brief overview for the Office for Health and Medical Research, where I work at the Ministry of Health. So we have strategic areas of focus. And as you can see, several of these have focused on the next generation antimicrobials, including Phage Australia, the Phage Regulation Working Group, Phage Manufacture, and we also look at GMP facilities as well to help translate these novel therapies into clinical practice in Australia. So starting with the Phage Regulation Working Group. So this was founded in 2022. And obviously interesting concept, working with phages being the natural organisms, and not being a traditional medicine or what we would think of as a biological. So our initial questions were, where does GMP start? How do we even think about moving these viruses from the sewerage, I guess, and getting them into a medicinal product to be able to administer to a human. So how phage should be classified by the Therapeutic Goods Administration, which regulatory pathway is most appropriate, what guidelines already exist, and what do we need to do to help our regulatory agency address the issues and help developers to manufacture these therapies. So in 2023, New South Wales demonstrated support for patient access to phage therapy, identifying that manufacturing capacity was a bottleneck for phage therapy, and invested 3.5 million in manufacturing, with limited facilities operational globally and the additional wait times for phage therapy to be imported. So this was obviously established at the Westmead precinct, where John and the team are, and that funding lasted for a couple of years. Sorry, this screen, there we go. So I said that this hasn't been a quick process, so I just did a bit of a timeline here to show the significant milestones and achievements in the development of bacteriophage as standard of care in Australia. So this shows extensive coordination between various different teams to achieve these milestones, from regulatory approvals to clinical trials and funding initiatives. So starting in 2017, John and the team used an intravenous phage cocktail approach to successfully treat 13 patients with Staph aureus associated sepsis. 2019, using a personalised phage therapy approach to successfully treat a 7-year-old child with Pseudomonas aeruginosa associated osteoarticular infection. Phage Australia was established in 2021 on the back of a federal grant that aim to professionalise phage therapy as a significant intervention for infectious diseases. And our office also invested in PhDs and early mid-career researchers to help build the phage workforce. As you can see, our regulatory working group started here in 2022 with our investigation more intensely into the GMP regulatory pathway starting in 2024, and taking us in 2025. We actually started working very, very closely with the Therapeutic Goods Administration and the Office for Gene Technology Regulators to decide on some safety statements of how we would actually produce phage, considering the safety markers, so that we can standardise that quality of phage manufacture throughout Australia, and to help the Therapeutics Good Administration decide exactly how they're going to register this therapy, so that we can use it as standard of care. So here are the guidelines that we've developed. So the Office for Health and Medical Research coordinated this national multidisciplinary modified Delphi process, which has been used to develop and refine a set of consensus statements based on the best practice evidence and expert opinion. So we used a Delphi approach because this is a well-known method used in healthcare to provide statements for experts to reach a consensus on a particular topic, especially when it's quite complex and we require collective intelligence, across research, across industry, across government, regulatory, production. So we needed quite a wide lens for this. And you can see on the right-hand side, here's the methodology that we used. So throughout this process, it's got several phases. So we had initial consultation with specialists where I then developed the proposed draft consensus statements on sourcing and preparation of phage, focusing on the GMP processes for medicinal phage. So we don't want to be putting any guidelines in place to restrict any general laboratory research for phage therapy. We're really focusing on that medicinal phage that's intended for human or veterinarian use. So once we had these draft consensus statements, the Delphi was begun. So we invited all 48 members of our regulatory working group to participate in this Delphi process. We ended up with 18 core attendees, I suppose, and contributors to this Delphi process. So during that first round, a red cap survey was distributed to everybody, which included all of those statements, and everybody could then anonymously respond to the statements, being able to say if they agreed with the statement, if they rejected the statement, and what the concerns were with the rejection for the statement, if that were the case. So bearing in mind, we were trying to build in the safety markers, so what the minimum requirement would be to meet that safety requirement to give phage to a human or animal. We then, I went through and evaluated all the responses and collated them together. And then the second phase of the Delphi was face-to-face workshop where all attendees attended, I suppose, either in person or online, because remembering this was a national validation process, so across the whole of Australia. And we went through each statement one by one with all the comments that have been received, and to collate consensus on those responses. So we began with 43 statements. We had 12 responses through the red cap survey. Only 10 statements were accepted as they were with no comments, and then we had to review 33 statements on the day, which was quite labour intensive, but quite interesting conversation. And then we came up with some finalised statements. So we now have 35 finalised statements that we're very happy with, and we're just about getting those ready for publishing now, which is quite exciting. And here's some of the team that came on the day. You might recognise some of the faces presenting for you today. Again, we had regulators here, so the Therapeutics Good Administration also attended the day. So we're really making sure that the statements that we developed are fit for purpose to help get these phage therapies regulated as soon as possible. So next phases, so the TGA being heavily involved with us, have since proposed a GMP exemption so that we continue to manufacture phage therapy within our hospital systems, allowing a 3-year exemption for small batch phage therapy productions, whilst all this regulation is being put into place, and the TGA designed their therapeutics good orders for that safe manufacture standardisation across Australia. Phage Australia has the ISO 9001 QMS certification, which is great. So they're providing that quality management system for safe preparation of phage therapy, and working on GMP scale up currently, because we do have a bit of a wait list in New South Wales at the moment for these very important therapies. And just to wind up, we don't just work on regulation, we also work on workforce preparedness, I suppose, to be helping, to deliver these therapies once they get to the hospital to our patients. So we've also worked with the Advanced Pharmacy Australia, which is the professional body here for pharmacy. And we've developed a pharmaceutical advanced therapeutic production credential to try and standardise that safe preparation as well. And here are the details for HMR. Thank you.
Thanks, Kerry. That's fantastic. I think it's just while I think Carina is getting ready to line up her slides. It's good to point out the value of the government interaction. I mean, from my point of view, working with Kerry and Julia and Laura at the OHMR has really made an enormous difference. I'm sure we're years further ahead than when we would have been otherwise without their support and all the innovative work that you've seen. So we might hand over to Carina, and Kerry gave a perfect segue, I think. So Carina.
You're on mute, Carina.
You there, Carina?
Sorry, can you hear me now?
Yes.
Yes, you're doing great.
Perfect.
And can you see my screen?
We see it.
Great. I can't see my screen, sorry. All right. So sorry about that. I'm going to talk about some of the important elements of a clinically relevant therapeutic phage biobank. So here at Westmead, we think it's really important to be fully integrated into the diagnostics and manufacturing workflows. And that means that as a biobank, we can be really data-driven. So we can look at things like which of our phages in the collection perform consistently well in diagnostics, and which of our phages purify well. And that way we can really prioritise our R&D in the biobank. We bank our phages in a 2-tier seed lot system, and that ensures that we are maintaining the integrity of our stocks. Phages need to be genomically and phenotypically characterised, and ideally host range needs to be tested on clinically relevant reference sets. We'd like to have a diversity of phages in our collection to cover that broad host range. Collaboration's been really key to establishing our biobank, but we are finding that all of the institutions that we all work for across the globe are becoming more concerned about IP and ownership issues. So as researchers, we really need to become better at understanding and managing IP. We just heard Kerry talk about the regulatory space here in Australia, and biobanks really need to be ready to adapt to that developing landscape. Here at Westmead, we have our phage therapy pipeline, including the biobank, is ISO 9001 certified. And we believe that really puts us in a good position to adapt to to those developing changes. In terms of infrastructure and resources, I've just listed some of the key elements here. Obviously, we need cold storage. Phages don't take up a great deal of space, but it is important to have some backup capability and an equipment management system that can monitor those fridges and freezers to make sure that we're maintaining temperature. It's really key to have access to high turnaround sequencing and bioinformatic analysis so that we can assess our phages for safety and purity. And another really key element is a database or a LIMS system. Here at Westmead, we really record just about everything that we can about our phages, starting off with where our phages have come from, where it was isolated, all of the biobanking details like the titer and the aliquot numbers and the usage, and any sorts of agreements that we have around our phages. all of those genomic characteristics, importantly, the safety assessment features and other things like taxonomy and morphotype. We also record every titer, every propagation that we do, every purification that we do, and all of our diagnostics data, all of our QA/QC for our batches, and also where each batch, each vial has gone. With all of this data that we amass every day, it means that we can analyze that data and use it to continually improve our processes as well. And so what have we got in our collection and how do our phages stack up? Being born out of a research laboratory environment, we do have over 500 phages in our fridge. But out of that, so far, we have 74 unique phages in the therapeutic collection. And that means that those phages have been sequenced and have met criteria for safety, for purity, and for uniqueness, and have been banked in that 2-tier seed lot system. We really focus our therapeutic collection on the 3 species that make up our highest referrals, so that's Staph, Pseud, and E. coli. And when we looked at the last 75 cases that we've performed susceptibility on, we were able to get a match in 65 of those cases. So looking at our 3 top species, we're actually getting a match in 80 to 90% of cases. We also recently tested our 3 staph phages on a reference set of 60 blood culture isolates that were collected around Australia, and we were able to get a match in 88% of those isolates. And so the question now remains, and what we're working on is what phages are we missing out in our collection to cover that 10 to 20% gap? And how often are we going to need to update our collection in order to maintain coverage over those clinically relevant strains that are circulating in the population? And that's it from me.
Thank you, Carina. That was fantastic. Thanks for a quick overview. I think Steph is going to take over. Steph Lynch has spoken before as well. So Steph's going to talk a little bit about the next step.
Thank you, John. Let me get back to my slides. You can see my slides okay? And I should put them in full screen, hey? All good?
Yeah, that's great.
Perfect, thank you. So as John mentioned at the start, I'll be talking about the feasibility aspect of phage therapy today. So this isn't about whether phage therapy works. It's about can we deliver the phages safely, reliably, and efficiently. And I guess one aspect to this is the lab operations and more specifically the phage matching or phage diagnostics, as well as the production. And so in this really brief talk, I'm going to be discussing 4 main questions that often come up when we're looking at the feasibility of our phage therapy pipeline. So the first one being, do we receive enough strains to match phages effectively? So this is just one case example that I'm going to talk through of the 203 cases that we've received strains for over the last 4 years. But in this example, so the case was referred in March last year. We received the first isolate soon after referral, and we matched 2 phages as we usually do. We try and aim for a 2-phage cocktail for each case, phage 1 and 2 that I've just called them here. Soon after, we received isolate 2. Luckily, the same 2 phages that we matched also matched isolate 2. Just before treatment, so the phages had already been signed out there with pharmacy ready for treatment. We did receive a 3rd isolate, and unfortunately the 2 phages that we did match to those first 2 isolates had no efficiency on that 3rd isolate. So for this patient, the treatment did start despite this finding. So on the flip side of this, we have had cases where we received 10 isolates all at once at the time of referral. We're able to match a 2-phage cocktail to all 10 of the strains. And so what I want to highlight here is that by receiving what we say like 3 to 5 isolates, recent isolates at the time of referral, we're able to more efficiently match phages against the whole batch of isolates. It also gives us a bigger picture of the susceptibility profiles that we're seeing. And by receiving them all at the same time, it impacts what treatment we're going to proceed with, as you can probably see here. And I guess what I'd like to put in front of all of these is ideally we want to have 3 to 5 isolates here in the lab for phage testing at the time of referral to capture the within patient strain diversity, with the idea of matching those phages to those current, not historical pathogen populations. So this is especially important where the populations shift over time and therefore phage susceptibility may differ. And then we're saying ideally aim for isolates that are less than 6 months old to ensure accurate and clinically relevant phage matching. And this is where patient selection and clinical microbiology is crucial for the efficiency in phage diagnostics. The other question that Carina has already covered a little bit is, do we have enough phages in the bank? So Carina has already proved that we have quite high matching rates with the banked phages that we do have for the isolates we are referred. But I wanted to touch on how many unique phages did we need to treat the patient. So the data that I'm going to present here is based on 45 treated patients. And if we assume that every patient gets 2 phages, as that's what we aim for, so 90 unique phages and 90 purified batches if we were doing it per patient. But what we were able to see is that of those 45 patients, we required 29 unique phages, so about a third of what we're expecting, and 44 unique purified batches, so about half of what we were intending. So what I want to highlight here is for the isolates that we were referred, we required a relatively small phage library in order to treat those 45. And this likely comes to, as Carina mentioned, focusing on key pathogens, so Staph, E. coli, and Pseudomonas. And what we've seen is within these key pathogens, we have a few what we call workhorse phages that can be used multiple times across different patients, especially in our Staphylococcus patients. So this is to highlight that phage usability obviously improves efficiency by we don't have to start from scratch and re-purify, and then we're leveraging those pre-characterized phages that we already know a lot about. And one thing I want to highlight here, because it's often a point of discussion about personalized versus large scale, and I think what is possible, or what I hope to show, is that scalability is possible even in personalized phage therapy, as the personalization can come down to the compounding step at the bedside. So having all of our phages large scale purified on the shelf, and then just picking personalized medicines to be put together at the bedside. So the other, I guess, question is about our diagnostics specifically. And as we know, there's no validated methods for phage susceptibility, and this is something that UCAST is working on. So I'm just highlighting what we work off in the interim. So do our EOP and kinetic results align? So for EOP, we generally use a threshold of greater than 0.1% to then proceed to kinetics, unless it's in like a part of a isolates from one patient, then the isolates may go ahead even if they have a low EOP. And hopefully what you can see here is that in 80% of the cases where we have a true EOP, so this is greater than 1% EOP obtained, we also have a true kinetics. And for kinetics, what we use is a mix of visual representation, so looking at the curves of the growth patterns, as well as area under the curve, just to have a quantitative measure. And so we are still investigating the concordance between EOP and kinetics for Pseudomonas and E. coli as our 2 other high referral pathogens, but we have seen a high concordance rate in Staph aureus, and this is a publication that's due to come out shortly. So just reiterating that in 80% of the cases where we have a positive EOP, we also see a positive kinetics result. But on the flip side, because this is based on treated patient data, we would have to do all the negatives as well to have those, I guess, positive and negative predictive values that we talk about. But these predictive values will one day be very important in efficient matching in terms of time and resourcing. So the last quick thing that I want to touch on is, can we meet those clinical timeframes? So this is obviously best case scenario, but I did put in brackets rare. So our quickest turnaround in terms of referral to treatment has been 7 days. This obviously requires that we have the purified product of the match phage on the shelf. So 1, we have a match, and 2, we have it purified, that we have enough doses on the shelf for the patient, that the hospital and the patient are actually ready for phage delivery. So comes down to governance and ethics and those kind of aspects. At the moment, our main wait time is sterility release, so a 5-day sterility. I know other places have a much longer sterility timeline, but this has occurred 3 times since 2021, all for Staph aureus, and arguably the important pathogen to be getting around in a tight turnaround. But more realistically, at our current capacity, we aim for around a 4 to 6 week from referral to treatment. This requires that we have the phage diversity to match the bacterial diversity that's coming in. So going back to Carina's points, so to improve these timelines, understanding the epidemiology and phage isolation is obviously going to be important. One of the aspects that also, I guess, feeds to the larger timelines is the purification. So the unpredictability or the inconsistency during purification, which again, Carina touched on, that we often use the ones that are purified well. And then obviously getting sites set up for phage administration is another big one. So again, coming back to that ethics and governance. So in this really brief talk, I hope I've mentioned some things that require further discussion on the feasibility, but I'll hand back to John and Holly for the safety side of things. Thank you.
Thanks, Steph. That's fantastic. So it's worth pointing out, when you say 7 days from phage on the shelf, you mean 7 days from high titer stock that then has to go to fill and finish in that week.
Exactly.
Yes. Thanks again, Steph. In the remaining time, we'd like to spend about 5 or 10 minutes on some of the downsides, I suppose, or the adverse events, and then allow a few minutes for questions. So thanks, Holly.
Great. Thanks, John. And they've been really fantastic talks. I've been taking notes as everyone's been talking as well. I hope you can see what I am showing. So I think we've touched on the standardized treatment and monitoring protocol for patients receiving phage therapy, which was developed by John Iredell and Ameneh Khatami. The primary outcome here is the safety and tolerability of phage therapy assessed 15 days after completion of therapy, with additional secondary safety outcomes and endpoints looking at any serious adverse event that has occurred, or an adverse event that is related to phage therapy up until 6 months post phage administration. And what we can say so far, obviously this is still an ongoing study, but what we have seen so far is that in a patient population who has a lot of comorbidities, we are seeing a number of unrelated serious adverse events, which have been 24 serious adverse events in 12 different patients. There's also been probably related non-serious adverse events, 4 in 2 patients, including fevers, hot flushes, tachycardia, rigors, and Bell's palsy in 1 patient. Along with this, there's a number of possibly related non-serious adverse events, which I will also touch on a bit further when we monitor for vital signs during phage therapy. There have been 2 probable and 2 possible serious adverse events that have been related to phage therapy, which includes 1 serious adverse reaction in 1 patient that had 2 episodes of liver enzyme elevation in a susceptible patient with a liver transplantation. There have also been 2 possibly related serious adverse events, one in a patient with a background of chronic kidney disease, developing acute and chronic renal failure, and another patient that developed a different strain of E. coli urinary tract infection in the context of a catheter used for installation of phages into the bladder. During the STAMP protocol, we monitor quite strictly the vital signs, including fever, any change in vital signs, any increased baseline pain at the site of infection, or any other signs of a possible inflammatory response. Most of our patients will have intravenous phage infusions, and we will monitor for these changes in vital signs at 15 minutes and 30 minutes post-administration. And what we can see is that these changes, you can see here in the proportion that is occurring per patient is actually quite common, including fever and any changes in vital signs. However, when we look individually at what vital sign is changed, we can see the most common occurrence is that of increased heart rate, decreased blood pressure, and increased blood pressure, all at rates of greater than 5% per patient course. Other signs of inflammatory responses, when broken up into signs, are all less than 5% in the patient cohort. And why are we so interested in these signs of inflammatory responses? It's because we have demonstrated this before. So this is a published case. Ameneh, Khatami, John Iredell and team described an inflammatory response with immune gene expression with a Yarix-Herxheimer type reaction where there was rapid bacterial lysis, which is detected with bacterial DNA-emia within the serum of a child with Pseudomonas osteomyelitis. The DNA-emia is then followed with amplification response of the phage also detected within the bloodstream. And transcriptomics revealed that there was upregulation in genes involved in the innate immune system at the time of these sustained sort of inflammatory response in this child. Under STAMP, we have also demonstrated quite a significant inflammatory response in a patient that had sequential phage therapy for a chronic E. coli post-pneumonectomy empyema. This termed infusion reaction after the phage treatment number 2. Initially thought to be related to an endotoxin reaction, was later discovered to be more likely related to rapid bacterial lysis and ongoing phage replication. We can also look at the temporal relationship, phage kinetics, with that of our serious adverse events that have occurred, including the case with liver enzyme derangement and the patient that had acute on chronic kidney disease. For the first case in a patient with cystic fibrosis and liver transplantation with Pseudomonas chronic lung infection, we can see that there were peaks in phage concentrations, both seen in the serum as well as the sputum, which correlated with the liver function test derangement. However, for the serum creatinine rising in case number 2, initially the rise occurred at the time of phage amplification, however, continued to rise despite cessation of the phage and then no phage detected within the bloodstream. So I will stop there because I'm sure there may be questions. Thank you.
Thanks, Holly. That was a fantastic summary of a lot of detail. So I'll hand back to our chairs, I think, Greg and Gina, and let you take us through to the end.
Thank you so much, Phage Australia in general. There's so much success that's happened, and you're leading the way of having a single site being able to do both the clinical side of thing and the laboratory side of thing, and now how that you're bringing together the regulatory muscle and working to get collaborating on key documents. You're essentially leading the way in. You're a 5-star baseball player, Phage Australia, if you know what that means. Maybe you like cricket or something to that effect, but I'll leave it at that. I think we're ready to take questions from the group. I think we also have opportunity to, both Gina and I will ask questions with the time remaining as well. Any questions? Gina, did you want to start?
Yeah, this really is so inspiring and fantastic, guys. This really is, blows us all away. And so thank you so much for presenting. My question has to do with, this level of national coordination that you've managed to achieve. For those of us working in more decentralized health systems, what kind of advice would you have for kind of making the case for governing bodies to make phage therapy a priority, that it's a common good, perhaps you could make that argument, for treating patients who otherwise don't have any other options? How did you make the case to the government to fund this and make it important?
Well, Gina, that's a big question. I guess I would point out that Steph and others led a bit of a consensus review of what people wanted, the prescribers, that is to say, what do the infectious diseases physicians think they need to do? And I think, Kerry, you might be the best position to make a comment on what the government thinks about this kind of stuff.
Right, thanks, John. I guess we're quite unique in New South Wales in particular for having an advanced therapeutics team, which was only developed 6, maybe 8 years ago, I think. Started as 1, there's now 4, nearly 5 of us. So we've just slowly expanded. We do a lot of horizon scanning and looking for these new therapies. And that has been really good. As you can see, there's been government support for these sorts of therapies. So I guess having this unique system and having this team looking for these therapies and trying to help support our local researchers. And I mean, obviously phage therapy and AMR, it's a global issue. So the stats speak for themselves, I guess. And then, yeah, being governed, I think just the specialists within our team, we just seem to have the right skill set to, connect with regulatory and, phage therapy being a hot topic, I guess, and there are some national experts as well that John, I mean, he's also internationally connected as well. That's just helped along the lines of, of demonstrating the importance, I suppose. So governments need to be thinking about advanced therapies, I think, and trying to get some specialists and some interest in that area. I think that's what you need.
And Kerry, how much do you think the prescriber survey and the health technology analysis and the health economics data that, in fact, you guys commissioned, how important do you think that was?
Definitely. I mean, to get health to support, you need to prove the economic component. So, doing those surveys showing that it is worth the investment is definitely what you need to get the money behind it, I guess, initially. And yeah, that's
great. I see we have a question from Dr. Dennis Oteno, PhD from South Africa, and please put yourself off mute and ask your question.
Great. Can you hear me?
Yes, we can, please.
Yes.
Great. So my question is to the phage, in phage bank, do they do a cocktail of the phages or it's just single phages for for the phages you work with, and do you have phages against MTB? Do you work also with MTB? And I have a question about phage and immunology. So
Can we,
when we
sorry,
use
I don't mean to
phages,
interrupt, but we're very interested in, and if it's okay, just try to take the 2 questions at a time and then see where that leaves us. Is this okay? If you want to write that 3rd question in the chat, maybe this is acceptable, please.
okay.
Thank you so much.
Thank you.
Maybe we'll direct that to Steph. I mean, when we started, we were, beggars can't be choosers. That was the case, wasn't it? We were often using single, but now I think, Steph, you try and combine,
Yeah,
but
that's
it's
right.
always
And
chosen, isn't it? Rather than
yeah,
an empirical tale.
exactly. And I think correct me if I'm wrong, but I think the question was also around the banking of it. So we bank them as individual phages, and then, as I mentioned, combine them as a cocktail at the bedside rather than storing them on the shelf as a cocktail as well. So that's the other thing that allows the personalization effect to choose and pick what you need for each patient. But you could have multiple on the shelf at the time. So I hope that answers the first part of your question. I'm trying to remember the second part.
It was NTM or Mycobacteria tuberculosis specifically for phages.
PB, I think we certainly don't have them. Graham Hatfull, I believe, is running a trial at the moment. I'm not exactly sure where, but certainly in an African nation or multiple African nations on the potential value of antimycobacterial phages or MTB. That's obviously a bit of a holy grail, but a very difficult one. So we can't really speak to that.
And not seeing any other questions. Dr. Ateno, you had an immunology question. We have time for that now.
There's a question in the chat too, Greg.
Sorry,
from
please, Gina, why don't you take that one?
Yeah, John, do you want to ask it yourself?
Yeah, thanks. Great talk, folks. It's wonderful to see all of your phenomenal efforts, and I miss being on site to chat with you about cases. My question was a little bit about the characteristics of the bacteria that you don't have phages for, and how is that informing kind of your hunt versus the potential to train, and because you had such a great platform for sequencing, are there any characteristics of those bacteria? And then the flip side would be, is there any evolution of changes with your bank in terms of, repeating that data to see what is working and what is not working? And the example would be in our CF patients, the almost all of our initial bank for Pseudomonas is not working for our patients post-modulator, and I'm just curious if you're also seeing that. Thank you.
Well, that's an interesting and complex question, John. So, Steph, I think it's fair to say that we are driven a bit by demand and limited a bit by capacity. So there are many of the questions you're asking, which I think all of the biologists would be wanting to go straight to, that we're just really unable to tackle completely at the moment. I think, Steph, it's fair to say that it is often the case that when we don't get a hit with our initial set, jumping into a huge extended search has got limited value, probably because of the nature of the resistance. Steph, did you want to comment on that or find fault with my response?
No, I think I completely agree. When I was reading the question in the chat, I also agreed about the CF Pseudomonas population that you obviously are seeing as well, because you may have seen on Carina's slide. So Pseudomonas is our highest referral, and it's the one that we do tend to not have a match for more often, especially in the CF population. So now sequencing all of those isolates and understanding why are we not getting a match? As you said, are they things that we can overcome with training, or are they internal defense systems that are going to be a bit hard to bypass? So I agree with that, John.
Yeah, John, your team has been a great leader in this space with not only, defining the right kinds of synergistic combinations, but also defining the right ways to attack these things. And it's a very interesting area to explore further, I think.
Yeah, we may have a chance to circle back about that, because we we could be informing kind of what's happening in the CF Pseudomonas community and kind of taking a look at your data, our data. That could be really informative so that'll be fun to consider. Yeah, thank you.
Okay, I think we have concluded our session from a time perspective today. For those who would like to come on video, here's a chance to give a thumbs up to Australia, doing a fantastic job coordinating a center, and this will be something that will be shared and put on the news. So we're going to go 1, 2, 3. Great job, everyone, and we'll see you Monday. Gina, any 1 or 2 words you want to mention about your talk on Monday?
Very difficult patient with chronic urinary tract infections with bladder reflux and a multi-drug resistant Pseudomonas.
Excellent.
Ouch.
So we're going to get into the the nut of the biofilm. I don't know what you want to call it. All the best, and we'll see you soon. Welcome back, GCPR. And for those who are going to be in Munich, we'll see you soon too. Bye for now.
Thanks, everyone.
Thanks, everyone.
Thanks, everyone. Bye.
Good job.